Hi, in this brief video, we're going to talk about three different ways an enzyme can be inhibited. In the first case, we're going to look at the case in which the Km, or Michaelis constant, is increased and the Vmax is unaffected. So here is a typical Michaelis-Menten enzyme kinetics curve where we are relating substrate concentration on the x-axis to initial reaction rate on the y-axis. And we see that at very low substrate concentrations we get a nearly proportional or linear increase in reaction rate as we increase substrate concentration. But at very high substrate concentrations, our enzyme activity, or initial reaction rate, no longer increases proportionally with substrate concentration because the enzyme is saturated. We call this maximal initial reaction rate, Vmax, and we call this number right here Km, or the Michaelis constant. Km is a specific substrate concentration at which the the initial reaction rate is equal to 1/2 Vmax. So to find Km on this graph, you would say here's Vmax, here is 1/2 Vmax, and then wherever this curve is present at 1/2 Vmax, we go down to the x-axis, and that is our Km value. So here is a graph of an uninhibited enzyme. If we have an inhibitor that is going to increase the Km but not change the Vmax, then we're going to go down to this x-axis and move our Km further to the right. We're not changing our Vmax, so this Vmax is going to remain the same. And because Vmax is unchanged, 1/2 Vmax is going to be the same value. And so now we know that this graph is going to pass through a point, the new curve is going to pass through a point in which the x value is this increased Km and the y value is the same unchanged 1/2 Vmax. So we come over here, and here is that point on this graph that is increased Km and 1/2 Vmax. And then we sketch this curve such that it now passes through this point. And we should go back and look at this graph and be able to convince ourselves that we are at the same Vmax. So look, both of these graphs plateau at the same height. So they both have the same maximal initial reaction rate. But this inhibited graph takes longer to reach that Vmax. So what I mean is it takes a higher amount of substrate to reach saturation. So at very, very high substrate concentrations, this enzyme is going to function similarly in the uninhibited and in the inhibited state. But remember, in our cells, we have substrate concentrations that are far below saturating. So in our cell, we might have a concentration that is somewhere around here on the x-axis. And so we can see that, let's just say we have this arbitrary substrate concentration where my pointer is. If we have this much substrate in the uninhibited state, this is the rate of substrate conversion to product. But in the case of the inhibitor, when the inhibitor is present and we have this much substrate, now we have this reduced rate of activity. And so this is what happens when we have an inhibitor that increases Km but does not change Vmax. This is characteristic of a competitive inhibitor. Competitive inhibitors will typically increase Km without affecting Vmax. Allosteric inhibitors could also function this way. So if I showed you this graph, you should be able to tell me this is consistent with either competitive or allosteric inhibition. Here's a second way we can inhibit enzymes. We can inhibit enzymes by decreasing the Vmax without changing the Km, or the Michaelis constant. So here again is an uninhibited enzyme kinetics curve. We see substrate concentration on the x-axis, initial reaction rate, or V, on the y-axis. This curve plateaus at a particular value on the y-axis, which is our maximal initial reaction rate. Here is the 1/2 maximal initial reaction rate. And we come over to where the curve is and then come down, and here is our Km. Now in this case, we are going to reduce our Vmax without changing Km. So now I've drawn a case where our Vmax is lower on the y-axis, so our new curve is going to plateau at this red line. Our Km is going to be unchanged. So this Km for the uninhibited enzyme catalyzed reaction is going to be the same Km, meaning the same concentration of substrate will get us to 1/2 Vmax. The difference is 1/2 Vmax is going to be a smaller number in the case of the inhibitor. So if we're reducing Vmax, we're also reducing 1/2 Vmax. But our curve is going to now pass through a point on this plot where the x-axis is the same value for Km. The y-axis is our now reduced 1/2 Vmax. And so this spot right here is where our curve is going to pass through. And there it is. So this inhibited curve is going to plateau at a lower rate. We have a reduced maximal initial reaction rate, but we have the same Km. The same substrate concentration is getting us to 1/2 Vmax. In the uninhibited case, 1/2 Vmax was a higher initial reaction rate than it is in the inhibited case. But in both cases, it is this particular substrate concentration that is getting us to the half maximal rate, whether that's the uninhibited or inhibited rate. Now remember, Km is essentially a reflection of the substrate's affinity. So in this case, where we are reducing Vmax but not changing Km, we're having very little, if any, effect on the affinity of this enzyme for the substrate. What we're doing is inhibiting the ability of the enzyme to convert substrate into product after they have bound to each other. And again, we can visualize how we're getting inhibition. So let's just pick an arbitrary substrate concentration, maybe right here on this x-axis. In the uninhibited state, this is our initial reaction rate. Whereas in the inhibited state, at that same concentration of substrate, we are actually getting a lower initial reaction rate. So it's a different way to inhibit an enzyme where we are now reducing Vmax without affecting Km. This is one way that allosteric inhibitors function. Finally, an inhibitor could do both decrease Vmax and increase Km. Let's look at that. Here is an uninhibited enzyme. This is the Michaelis-Menten kinetics curve for an enzyme. Substrate concentration on the x-axis, initial reaction rate, or V, on the y-axis. The curve plateaus at a particular value, Vmax. This is the maximal initial reaction rate. At very high substrate concentrations, our enzyme approaches Vmax because the enzyme is saturated. Virtually every enzyme molecule is busy converting substrate to product. At very low substrate concentrations, we get a nearly linear increase in initial reaction rate with increasing substrate concentration. And Km, again, is just a value on the x-axis. It is a particular substrate concentration at which the initial reaction rate equals 1/2 Vmax. Now in this case, we are going to both reduce Vmax and increase Km. So here's going to be our reduced Vmax. Here is our increased Km. If this is our reduced Vmax, our 1/2 Vmax is also going to be reduced. So now 1/2 Vmax is going to be at a lower point on the y-axis than 1/2 Vmax was for the uninhibited enzyme. And now we can plot at least one point that this curve is going to pass through. We know that the curve is going to pass through this point on the graph, that is the intersection of Km and 1/2 Vmax. And now we can draw this curve and show this curve moving through that point, plateauing at a lower level. Again, we look at this curve, we see that our Vmax is reduced. So even when the enzyme is saturated, we are going to have a smaller maximal initial reaction rate. And our Km is increased, which means it's going to take us more substrate to reach 1/2 of the new reduced Vmax. So even though the 1/2 Vmax is reduced in the inhibited state, it takes even more substrate to get to that reduced 1/2 Vmax than it would have taken us to get to 1/2 Vmax of the uninhibited enzyme. And so this is another way that allosteric inhibitors can regulate or inhibit enzymes. And again, we can pick an arbitrary value on the x-axis, say somewhere down here, this particular substrate concentration. We see in the uninhibited state, we get this amount of activity, this initial reaction rate. Whereas at that same concentration, the inhibited enzyme has a much smaller value on the y-axis, a much smaller V, a much smaller initial reaction rate. The enzyme is inhibited. So what we've done in the last few minutes is look at 3 different ways to inhibit an enzyme: reducing Vmax, increasing Km, or both. You should also be able to use these same strategies to draw the ways in which an allosteric activator might increase enzyme activity. You can imagine that enzyme activity could be activated by increasing Vmax, reducing Km, or both increasing Vmax and reducing Km.